Oxidized LDL particles can actually change how macrophages express their genes. If this is not a familiar concept, let me explain. The cells of the human body all possess the same genes. The difference is which ones they use, which ones they express. And this can change from cell type to cell type, and this can change over time in one particular cell. A major process in the regulation of genes is the binding of transcription factors to DNA which turn on the genes. No human genes are active until the appropriate proteins bind to the beginnings of the genes, regions known as promoters and others, which then turn on transcription. These proteins are known as transcription factors required to turn on genes. There are groups or families of transcription factors. The estrogen receptor, which binds to its hormone estrogen in the cytoplasm and then travels to the nucleus where it binds to DNA and turns on genes. This is a nuclear receptor, that is the name of the family. Another gene in the nuclear receptor gene family is the peroxisome proliferator activated receptor gamma, or PPAR gamma. It is a nuclear receptor which binds with another one, the retinoic acid receptor, to form a heterodimer, two proteins stuck together which are different. But this needs to be activated, binding to something like the estrogen hormone, the way the estrogen receptor must bind to something. It turns out it can bind to oxidized metabolites from LDL particles. So no metabolites in free LDL particles can activate this nuclear receptor transcription factor. But one of the fatty acids, linolenic acid, can be converted into two metabolites which can bind to the PPAR gamma nuclear receptor and allow it then to turn on transcription of a number of genes. As a result, this macrophage will express different proteins after ingesting oxidized LDL particles, because metabolites from the oxidized LDL particles will bind to a nuclear receptor, go to the nucleus where DNA is found, and this will change the activity of genes. These genes include inflammatory signals, which may bring more macrophages to the plaque, and scavenger receptors, which may encourage the cell to take up more oxidized LDL particles. And so this can act as a negative feedback loop, growing ever bigger. A final interesting note: about 3% of the genome, the DNA in the cell, codes for protein, and so these genes are therefore known as the proteome, 3% of the genome. About half of the genome, however, is used and transcribed into RNA and is known as the transcriptome. Most of these RNA molecules do not code for protein, and it is now evident that there are a broad spectrum of functions that non-coding RNAs perform within cells. It is known that in many disease states, such as heart disease, there is a dysregulation of these non-coding RNAs and their expression. And it is known that oxidized LDL particles will change the expression patterns of some non-coding RNAs, including both small microRNAs and the much longer link RNAs. This is an area of active research.